All right, so I figured I would show you how to do a virtual fly lab cross. We're going to be doing a series of these. Usually I do 4 or 5 of these in genetics class. We're going to do 3. And they illustrate some basic concepts of genetics. And they can be kind of fun. And you get, I think 3 is plenty. I think you do a couple, you get the hang of it, etc. And so, the information for doing this is on the course web. And so you can click here to go to the Virtual Fly Lab website. We'll do that in a minute. And if you click on here, it gives you all the files that you need for doing these things. You can also get that information down at the bottom, and I put it all in here. So right now we're going to do instructions for Virtual Fly Lab assignment. And I'm going to follow those instructions for the most part. I'm going to show you those real quick. And so this is the document that tells you what to do. And I'm essentially going to do this. And I may or may not fill out the entire thing, but I can show you, I can get you started. So for this thing, you're always going to get a series of mutants. And in this case, you're going to get 2 different mutants, mutant 1 and mutant 2. And the first thing that's important is that you have to cross the mutant 1 female across to a wild type male. Eventually, we're going to cross mutant 1 to mutant 2, but we don't want to do that first. Students always jump right to that, and then it takes away from the sort of systematic nature of it, and it's harder to figure out what's going on. So we have to figure out what's going on with mutant 1 and mutant 2 by themselves first. So mutant 1 crossed to wild type, mutant 2 crossed to wild type, then mutant 1 crossed to mutant 2. All right, so that's what we're going to do. So I'm going to move that, and I'm going to go to the Virtual Fly Lab website. And, well, first of all, I'm going to go, let's see. I'm going to go to the VFL 1 mutant sets. And I'm going to do set 1. And that showed up over here. I got another screen going on over on the side there. So set 1 is A, P, and E. Okay, whatever that means. I don't know what that means. Let's get rid of that. And list of all VFL mutants. So let's find out what A, P, and E means. So if I open that document, there's AP, that means Apterous, and that's a wing size mutant. E is ebony, and that's body color. So you just have to remember that. And there's all these different mutants. So AP and E. So then we're going to go to the Virtual Fly Lab website. We're just going to enter as a guest. And we get this screen here, which shows us how to do Virtual Fly Lab. There's a few things. We can go to the computer and order some flies. We can put those flies in the incubator. We can look at them under the microscope. Then when we're done, we can throw them away. Under options, you can get rid of a bunch of sounds and transitions and things like that. So I'll do it for a while with these things on, and then I'll turn them off. We're not going to use any of this other stuff. We're just going to do our own little report. So let's see. Why don't I download one of those? So let's go to VFL record sheet. And so I've got that. And I'm going to keep that going on the side. And I'll fill that in as the time comes. So, all right, so I might as well put in my name. And let's see, what's my name? Ken Seville. The first one was AP, right? So that's the phenotype is Apterous. I'm going to cross that to wild type. So wild type is just normal. So Apterous, that actually means no wings. So that's the phenotype. Wild type just means normal wings. All right, so let's put that aside. So now I need to cross wild type to, or Apterous to wild type. So I have to go to the store and order some flies, and I want to make my female, and I wanted her to be Apterous. And Apterous was wing something or other. I think it was wing size. There's Apterous. You can see there's no wings. Here's wild type with wings, Apterous with no wings. Click on Apterous. Now my female doesn't have any wings. I'm going to put her in the cart, and then I'm going to design my male. I want my male to be wild type, so I'll just pick him the way he is, put him in the cart. Then go to the cart. I can check out. I say, yes, I really, really want to check out. And then I got my flies. And when you're just getting used to it, it's kind of nice because these little things pop up to tell you what to do. Put my flies in the incubator. This is awfully loud. Let's turn that down a little bit. Hopefully that's not so loud for you guys. And then I want to click on that and mate them. Takes 10 days to 2 weeks for flies to mate and give you babies. Here come the babies. Lots of babies. So now I want to take these things and look at them under the scope. Oh, I guess I got to open these up. Click on that. And it's going to put them under the microscope. And I can look at them. And we have to sort them. And this is really how you sort fruit flies. You take a little paintbrush, and so you have to knock them out with something. Get a paintbrush. And if I hover over this, it tells me what I got. I got my female or wild type. Over here we got male wild type. So plus always means wild type, no matter what trait you're talking about. If I look up over here, it tells me that I got 598 wild type and 623 wild type males. So females, males. All right, so that we could send the data to the computer to take a look at it and analyze the results. And when we see the males and females are pretty close, and this is always going to be the case unless we think that there's something going on with sex, like if it's sex-linked or something. But if we don't think so we can just ignore the sex. So a simple way to say this is we had all wild type. All right, so let's, and we can even sometimes I lose my mouse when I'm using two screens. All right, so we want this result. So I've got this thing called snipping tool. If you want to find snipping tool, go under here and type in snipping tool. And say, oh, there's snipping tool. And I saved it on my little bar here. So what snipping tool allows you to do is just take whatever you want, copy it, and then you go up to edit, copy, and I'm going to go to my record sheet. And so my F1 cross, I can paste my results right here. And I can even write down here the results of the F1 cross were all wild type. And I can say males and females. So there's nothing different about sex at this point. So what does that tell us? So apparent phenotypic ratio, I guess I could put this down here, it'd be better. So the ratio is all wild type basically. All right, I can give myself a little. So what can I conclude from that? So I crossed Apterous to wild type, and I got all wild type. So Apterous crossed to wild type, gave me all wild type. That tells me that Apterous is recessive to wild type. Now there are a few rules, and I put those in somewhere, that when you buy flies from the fly places, they're always going to be homozygous. So that means we had, so that tells us that maybe the cross was like this. Oops. Word's weird. Sometimes I wanted that to be two little a's. So that should make sense, right? Apterous is recessive, it's homozygous for 2 little a's. Wild type would be dominant, you get 2 big a's. Then you get heterozygous here. So once we know that, now what we're going to do is we're going to cross 2 of those together. Big A, little a, cross to big A, little a. What would you get? Now you should be able to predict that. Even if you haven't taken genetics yet, you should know that that's a particular kind of monohybrid cross. Right? But rather than predicting it, we'll go ahead and do the cross. So we're going to take our heterozygous, we want the phenotypes here, our heterozygous ones, but they're wild type, because these are the F1s and everything was wild type. We're going to cross 2 wild types together, and then let's see what we get. So we're going to go back here, return to the menu, return to the lab, take a look at our flies again. Now I'm going to click on that, and I want to use that female in a new mating. So now I'm taking the F1, use a new mating, and then this is the part I always forget. Now you have to zoom back out, and now you can go to the other pile, grab a male, and this is pretty much how you set up a cross. Use that in a new mating. Typically you'd use a couple males and maybe 3 or 4 females. Zoom out, return to the lab. Now I've got this thing. I got a new mating jar with my wild type female, my wild type male, but these are F1 wild types, right? So they should both be heterozygous. It won't take long for that buzzing noise to get annoying. So I'm pretty sure after this we're turning off our little noises. All right, so now let's check it out. I don't know why they put the bottle right on the computer. Now make sure you get the right one. So that's our first cross, Apterous female to wild type male. This is our F1 cross in cross number 2. Pick that. Knock them out with ether. We usually use carbon dioxide. Put them in there and sort them. Where's my sort button? All right, now I got a bunch of piles. If I look up here, I have 143 Apterous females, 487 wild type females, 125 Apterous males, 460 wild type males. So we're seeing similar results between the two. So if we look at the female results, we got 480 something to 140 something, 460 to 125. So I think we'll be able to ignore the sex for that. So we'll send the data to the computer, analyze the results, and we basically have pretty close numbers. I guess it's kind of hard to tell for sure if they're exactly close enough to ignore sex, but there's no evidence of sex linkage is basically what we're looking for here. So if even though you don't know what sex linkage is yet necessarily, I mean, I'm sure you've seen it before, you would see an obvious difference between males and females at this point. So we're going to ignore the sex, and probably if there's a real difference, it won't let you ignore the sex. So we'll have to see what's going on. So what this tells us is we've got 947 wild type to 268 Apterous. Now if you think about your basic Mendelian genetics, what we had here was a F1 of 2 heterozygotes, so it's a monohybrid cross. So we expect a 3-to-1 ratio of wild type to Apterous because wild type is dominant to Apterous. Well, let's test that out. So we think there's a 3-to-1 ratio. So we can do a chi-square test, and we can enter a hypothesis and test to see if it's right. Now, when you really do chi-square, you would actually use numbers that are based on the total that you counted, which is 1,200 or something. But what we're going to do is say our hypothesis is 3. I guess we can get rid of that. Say our hypothesis is 3 to 1. And so it will do the calculations for you. Now, that's one thing I don't know that there's any way around it, but this is somewhat misleading because when you actually do a chi-square on paper, you would not compare 947 to 3. But what this does is it calculates, if I count 1,200 and I expect 3/4 of them to be this, it would actually calculate the number that you would expect. But with this program, we can just sort of quickly test our hypothesis. So we test the hypothesis. It gives us this level of significance, which is low. And I think this thing usually tells you somewhere. Well, I thought it actually told you whether to accept or reject. And I'm not seeing that. So here's our 1200 expected. So our chi-square value is this. Degrees of freedom is this. And this is our level of significance. Okay, down here it says, if it is less than 0.05, it suggests that your hypothesis is unlikely. Well, that's weird. Because that would suggest that because it's 0.01, that would suggest that we reject our hypothesis. Well, isn't that interesting? Let's try that again. Should be 3 of these guys. So 1 of these guys. Test the hypothesis. 0.0179. Look at that. That would say that we should reject our hypothesis that it's a 3-to-1 ratio. So that's telling us what is the probability that this difference that we see from our predicted ratio, what is the probability that that difference is due to chance? This is saying there's a 1.7% probability that that difference from what we expect. So this is what we expect, 911 and 303. And this is significantly different from that. So that's weird. So we would actually have to reject the hypothesis. I didn't expect that. For this particular gene, it's just supposed to be a regular 3-to-1 ratio thing. But that's our result. So that's the way it goes. So let's go back to our record sheet. And we'll go. I should have collected those results. That's okay. We can just grab these results. I mean, these tell us everything we need to know. So we'll go back to our snipping tool, grab all this stuff, and that tells us everything you know. Copy it. So we can get rid of this thing. Go back to our virtual fly record sheet. Paste that in there. All right, so using data from the table indicate the phenotypic ratio. Say, well, I hypothesized a 3-to-1 ratio, but this ratio was rejected. So we can go back and test a different one. So let's enter a new hypothesis. Let's try 4-to-1. Rejected that one too. It's kind of weird. So we'll do another one and see what happens. So hopefully you won't get that. I mean, if it's rejected, there's not much you can do about it. So we can go analyze those results later. So let's check out the other one. So let's return to the main menu. Return to the lab. Now we're going to throw all those away because we're going to do another cross. So I'm going to do this pretty quick. So now I'm going to turn off these sounds and transitions and the directions because I know how to do it, or at least I think I do. And now we'll do the quick version. Order some flies. If you remember, the other fly we wanted was ebony, which was a body color. So we're going to buy a female ebony and a wild type male. And so you can see that's a darker female, wild type male. Check out. Yep, really want to check out. Unpack my box. Let them incubate. Sort them. wild type, wild type. Okay, send the data. You could probably you don't have to necessarily send this data to the computer and paste it in. You could just go here and say you would want a new we would want a new cross. So here we would write down our chi-square statistic and p-value from up here, and down here we would write our we would analyze our results. So I'm going to save this as my VFL1 cross 1 record sheet. So we'll save that. And then I'm going to open up a new one. So I think I'll have to go back to the course web, get another record sheet, and this is still me. Now we're doing ebony. Ebony female cross to wild type male. Alright, so let's go back and do that. And we got, and so I could just write in here my results. And you could write in the numbers or paste in the numbers, either way. Conclusions from that: ebony is recessive to wild type, and we actually know, even though we haven't covered it, we know it's not sex linked at this point. And I could just, the reason that we do this cross this way, if you took an ebony female cross to a wild type male and it was sex linked, you'd be able to tell that already. So if you don't see anything suggesting that one sex, so basically what you would see is all the males would be ebony if it was sex linked. So as long as you're not seeing any difference between the sexes in this first cross, then you know it's not sex linked. This, but that's only if you use the mutant female to begin with. So that's a little too much information, but that's how it is for now. So now we just want to mate some of these guys together. Let's take these females, using a new mating, zoom out. I always forget to zoom out. Check that male, using a new mating. Sounds good. Zoom out, return to the lab. Here's our new mating, our F1 wild type. All are wild type female and male, but they should be heterozygous for ebony. Mate them. It'd be nice if crosses went that fast. Analyze them, sort them. Got some kind of differences going on here. We've got 477 wild type, 136. So it looks to me like we can ignore the sex. We'll send the data to the computer. Analyze results. I would ignore the sex. Kind of looks, I don't know if that's going to be a 3-to-1 ratio or not. Our last one we got we got about 1,200 flies, but let's see. Do a chi-square analysis. You can wait to copy your results until you do all this analysis. 3, 1, test it. Now this says, so down here it says if it's less than 0.05, we're going to reject our hypothesis. Our hypothesis is that we have a 3-to-1 ratio. Our level of significance, or our p-value, in this case is greater than 0.05. So 0.259 is way greater than 0.05. So that means we do not reject our hypothesis, because this is telling us that the probability that the difference we see between observed and expected. So here's our observed is 926. Here's our expected is 909. So this is based on this 3-to-1 ratio. If we count 1,212 flies, if it's a 3-to-1 ratio, we expect 909 to be this and 303 to be this. What we saw was 926 and 286. So are these numbers significantly different than our expected numbers? Do a quick chi-square value, which is this formula. You add those up. You get a chi-square statistic, and we end up with a level of significance of 0.25, which is greater than 0.05. We'll go over chi-square more formally later, but right now we just want to know whether we reject it or not. If it's greater than 0.05, you do not reject. Okay, so it tells us not to reject that hypothesis. So that means it's behaving like a 3-to-1 ratio. Let's grab those results. Well, I don't want the snipping tool to be part of the picture. Get out of there. So we'll grab that. Control C to copy it. Go down to our results. Ebony cross to wild type. Using the F1 flies, do another one, paste our table here. Whoops. There's our table. Alright, so let's cruise down. Apparent phenotypic ratio looks like 3-to-1. But that's not good enough. We have to say 3 wild type to 1 ebony. We could write down those chi-square statistics. Conclusion. Looks like ebony is recessive to wild type and exhibits, let's say, inheritance typical of a monohybrid cross. All right, so down here it says rewrite the crosses with the genotypes in there. So we had an ebony female, we had a wild type male. So let's put in some genotypes. From our crosses now, we think that ebony's recessive. So that means it gets 2 little e's. Wild type gets 2 big e's. The F1, we had a big E little e, cross to a big E little e. Oops, sorry, we had wild type. Wild type, cross to wild type. Our genotypes were big E little e, cross to big E little e. Our F2 results, we basically got a 3 wild type to 1 ebony. So the genotype of that is going to be big E or big E little e. So we don't know which. Do a Punnett square and you'll be able to tell. That's the same as saying big E blank, because that blank could be either a big E or a little e. If ebony is recessive and we have ebony flies, they have to be little e, little e. That has to be their genotype. Overall conclusions, so there's not much going on here. So we only had 1 mutant. We didn't, so down here it says if we had 2 mutants, is there anything else going on? So for this one, I mean, it's kind of restating the obvious, but ebony is recessive to wild type, shows typical Mendelian inheritance for 1 gene. In other words, it was a monohybrid cross. Nothing else seems to be going on. Now we could say, if we were going to write the results, so we could say virtual fly lab 1 cross 2. For the Apterus one, we did see something weird. We don't know what it is, but it didn't look like a 3-to-1 ratio. Not quite sure what to do with that, to be honest with you. And it's not supposed to be weird like that, but hey, that's life in the lab, even in the virtual lab apparently. All right, so now let's do the next one. Oh no, I don't need to save that. So let's do one more cross. Return to the lab. Analyze results. No, we don't want to analyze results. We want to return to the menu. Return to the lab. Let's get rid of these guys. Because if you don't get rid of them, it gets too confusing because you have too many flies around. Now we're going to cross Apterous to Ebony, and in the instructions it says cross mutant 1 female to a mutant 2 male. So our mutant 1 was Apterous. That was a wing size, I believe. So Apterous female. Now with a male, we're going to make Ebony. Now everybody wants to do this immediately, not eye color, body color. But this is cross 3, so don't do this until you get to cross 3. You could do it earlier, but if you get something weird with either your mutants to begin with, you want to know that before you try to analyze these results. Unpack them. Isn't this better without all the sounds? Check them out. Sort them. We got all wild type, right? So when we're doing 2 genes at a time, we're essentially asking, when we do both genes at the same time, do they behave the same way as they did when you did them by themselves? And so we'll analyze that a little bit further. So right now we got wild type for both of these things. We don't need to send the data to the computer. We could just write down that they're all wild type, or you could send it if you want and copy the table. Uh-oh, went too far. I want to grab one of these males, use a new mating, okay. Zoom out. Return to the lab. So this is our mating jar. So if I did it right, then we should have the F1s in here. So this is a female F1 that's wild type, female or male that's wild type. Click on it, do the cross. Sort them, and we get a whole bunch of things. Males and females. Apterous, 126 females, 101 males, 339 wild type females, 300 wild type males. It's pretty much looking, so for Ebony by itself, 109 to 119, Apterous and Ebony, 39 to 38. So there doesn't seem to be a difference between males and females. So we're going to analyze these results and ignore sex. All right. So now let's assume that the Apterous gene gave us a 3-to-1 ratio, even though it didn't, but it was supposed to. If Ebony also gave us a 3-to-1 ratio, that means that both of those guys are behaving like normal Mendelian traits and giving us a 3-to-1 ratio in a monohybrid cross. So when we cross them together, they should both give us a 3-to-1 ratio by themselves, but then when we combine them, that turns into a 9 to 3 to 3-to-1 ratio. Okay. So is this a 9 to 3 to 3-to-1 ratio? Let's test it out. 9, 3, 3, 1. And if you think about it, so if this is 1 and this is 3, then this number, this 227, should be about 3 times that. And then these guys should be about the same. And then 639 should be about, so that looks to me like what's going on. Let's test it. Super high level of significance. This is a 70% probability that the difference we saw was due to chance. In other words, this is much greater than 0.05, so we do not reject our hypothesis. If it's less than 0.05, we would reject our hypothesis. So this tells us that it probably is a 9 to 3 to 3-to-1 ratio. So before we forget, Let's snip it. Copy it. I lost my record sheet. Hold on a second. Let's see, I guess I need a new record sheet. So let's go back here. Get a new VFL record sheet. So now we took an apterous female, crossed it to a wild type male, got all wild type, could fill in your conclusions up there. Well, we'll come back to that. Cross wild type female, cross to wild type male, that was our F1. F1. Paste in our results. There's our results. 9331. Apparent phenotypic ratio 9 wild type, 3 apterous, 3 ebony, 1 apterous and ebony. Chi-square statistic. Did I get that? I did not copy that, so we'd have to go back and get that. So let's just do that. We can even just snip that out if you'd like. We'll just paste those numbers in. That's fine. And that was in there. All right, so p-value and all that stuff. So the p-value is the same as level of significance. So 0.7, conclusion, do not reject 9, 3, 3, 1 hypothesis. All right, so now let's write it out. So when we do the crosses, we're actually, we're doing everything based on phenotypes. But once we do the crosses and we think we know what's going on, we can convert those, or we can conclude from that, or infer from those crosses, what the genotypes are. So here's where it gets tough. Pay attention. So we know this is apterous, female, and it was a wild type male. But now we're dealing with 2 genes at the same time. So we have to put both genes in so we can follow what's going on. The way we set up the cross, this apterous female, this is the really hard part. The apterous female is wild type for ebony. And all of these flies will be homozygous if they can be. This one is wild type for apterous, but mutant for ebony. We're keeping track of 2 genes at the same time. Once again, I did this. Well, this actually goes up here. All right. So then our phenotypes here were wild type. Wild type. And what are our genotypes? Well, our genotypes, we have to keep both genes in mind. So if we just think about the apterous cross, it was 2 little a's crossed to 2 big A's, which means we get a big A and a little a. For ebony, and we usually just put the big letter first, and then big E crossed to little e is big E little e. And that's what we call a dihybrid. It's heterozygous for 2 genes instead of just 1 gene. a monohybrid, it would have been heterozygous. So when we did the other crosses, big A, little a, that's a monohybrid. Big E, little e, that's a monohybrid. Big A, little e, that's a dihybrid. F2 results, we got a 9. You can do this any way you want. 9, 3, 3, 1. Phenotype was wild type. Apterous. Ebony. Apterous and ebony. Now the genotypes, there's a lot of different genotypes, but what we know about this is it had a big A, and then that big A could have a big A. The second allele could be a big A or a little a. So it could be, the genotype could be big A, or big A, little a. So we just do this. Same with ebony. All we know is it has a big E. We don't know which, and you get all different kinds. You get big A, big E, big E, big A, big E, little e. You get all kinds. Apterous, that means it's mutant for apterous, but ebony is wild type, so it has to have at least one big E. Ebony means it's wild type for apterous, but it's mutant for ebony. Apterous and ebony. AAE. Overall conclusion. So this is just giving you some introductions, and down here, apterous is recessive to wild type. In this case, it's looking like, and we can just say shows normal Mendelian inheritance. So apterous behaved normally in this cross. Ebony's the same thing. Let me just copy that. Say ebony. So both genes by themselves behave just like a normal monohybrid cross. For two or more mutants involved in a cross, do they exhibit independent assortment? How do you know this? Well, what independent assortment means is if you cross two different genes or two different traits, they give you the same results they would give by themselves. In other words, apterous is 3-to-1, ebony is 3-to-1. When you put them together, you get 9-to-3 to 3-to-1. Because what we get is 3/4 wild type and 1/4 apterous for one cross. For the other cross, we got 3/4 wild type, 1/4 ebony. All right, because that's what we saw when we did the crosses by themselves. So if we look at both of them at the same time, how many would we expect to be wild type for both traits? 3/4 times 3/4. We expect 9/16 wild type. We expect 3/16, so 3/4 wild type for apterous, and 1/4 mutant for ebony. Let's see how we did it up here. So we did apterous first. So 1/4 mutant for apterous, 3/4 wild type for ebony is 3/16 just apterous. And just to make life confusing, if it's wild type for the other trait, we just don't write it down. If we don't write it, it means it's wild type. So you got to write something here for the first one. So 3/16 ebony, that's what they look like. They just look ebony, but they have wild type wings. And 1/16 apterous and ebony. So what that means is 9-3-3-1. 9331 means independent assortment. You only get a 9 to 3 to 3 to 1 ratio if there's independent assortment. In fact, independent assortment, this is the definition of independent assortment. That if you get 3 to 1 for one cross and 3 to 1 for the other cross, if you do both those crosses at the same time in the same flies, you'll get a 9 to 3 to 3 to 1. Notice you have 9 wild type to 3 Apterus. That's 3 to 1 wild type to Apterus. You get 9 wild type to 3 Ebony. That's 9 or 3 to 1 wild type to Ebony. And this, if you did a Punnett square, it comes out the same way. But what we're trying to get away from is Punnett squares and multiplying probabilities together. Anything else going on? Nope. Now, if we saw something different than this, that might suggest that the genes do not show independent assortment. And then maybe there's something else going on. Or maybe we didn't get a 3 to 1, maybe there's some kind of genetic interaction going on. Rather than getting 9 to 3 to 3 to 1, we get 9 to 3 to 4 or 12 to 3 to 1. But it's some kind of version of this. So, yes, independent assortment, because the dihybrid cross gave a 9-3-3-1 ratio, which is, we can say diagnostic, but I'll say indicative of independent assortment. Cool enough. Save that. Saving is good. Virtual Fly Lab 1 cross 3. All right. I probably should have given you one more thing, but I'm going to show it to you because I think it's important. All right, let's do one more. It's going to turn into somewhat of a long video, but I want to show you something else anyways. Turn to the lab. Let's start over. So let's do this cross in a slightly different way. Same cross. Order some flies. And let's make our female, we could do the female or the male. We're going to give her Apterus wings and ebony body color. Put her in the cart. And the male will keep wild type. Put him in the cart. So this is essentially doing the same cross, but we're just starting out with both mutants in the same fly, and then we're going to cross them together and get an F1. These genes aren't sex-linked, so it doesn't matter that they're both in the female. And also, they're not linked, so it doesn't matter where they start. But we won't worry about that for now. So let's unpack them, mate them, check them out, sort them. We got all wild type again. So that's groovy. So what I want to do now is I want to do a test cross. So let's just take this male. No, let's take a female. Take a female. Use her in a new mating. Now rather than using this male, if I use that male, that would be a dihybrid cross again, because I would be taking, this one's heterozygous for Apterus and Ebony, this is heterozygous for Apterus and Ebony. But I actually want to go back and back cross it. So back cross is when you go back to one of the parents. And I'm going to back cross it to this female. And that makes that a test cross. Let's see. Oh, so I think, yeah, so we're going to have to replace that female. And then I'm going to have to go back in here and get a male. And so this is just kind of a weird thing. So let's see how our, do they put them in there? Let's see. So we got a new mate. So we got our female that's Apterus and Ebony, and the male is wild type. But if I did it right, that male is an F1 male that's heterozygous for both genes. Okay. A little bit goofy, but that's the way it is. So let's mate them. Mating 2. Now, what do we expect? Before we do this, let's see what we expect. We'll run. Let's see what we're doing here. We'll just use PowerPoint, a blank PowerPoint here. And all right, let's see. Okay, so these ink tools. All right, so let me just grab them. We'll just, for some reason I like blue. So what did we just do? For some reason I can't see it. All right, so now we're taking an Apterus and ebony female. Wow, this is something. Let me make this a little bit thicker. So Apterus, ebony female, cross to wild type male, and we got all wild type. So we know from previous crosses that this is recessive, and this is recessive. This is wild type. And then this is big A, big E, little e. All right, same thing that we did before. But now, rather than crossing it to another big A, big E, little e, I went back up to here to one of the parents and did a back cross and used the mutants. So now we're taking little a, little e, little e. What do we expect? Well, we expect this guy to give us these kinds of gametes. Big A, big E, big A, little e, little a, big E, little a, little e. We could do some kind of squiggly Punnett square. This guy can only give us a little a and a little e. So what we get down here, I'll spread out a little bit. We can get a big A and a little a big E and a little e. We can get a big A and a little a and two little e's. We can get a little a and a little a and a big E and a little e. Or we can get A, E, E. But now, rather than getting 9 to 3 to 3 to 1, I don't know why this pen keeps disappearing. There it is. We're going to get 1 to 1 to 1 to 1. Because this is a test cross. And a test cross, so the deal with the test cross is instead of using a dihybrid here, we're using something that's heterozygous or homozygous for both genes. Two little A's, two little E's. So that's the cross we're doing now. All right, so that was a little sloppy. I need to find something better to write on, but let's sort it out and see if that's what we get. Whoa, there's lots of stuff going on. So let's send it to the computer. We know from our previous crosses that these are not sex-linked, so we can do this. Do we get a 1 to 1 to 1 to 1 ratio? Let's test it out. 1 to 1 to 1 to 1. Test the hypothesis. Super high level of significance, or p-value of 0.8. That means we do not reject our hypothesis. That means that that test cross for two genes did show a 1-to-1-to-1-to-1 ratio. So that's something I need to add to this first virtual fly lab, is to, after you do this, to go and do a test cross. I did a little bit fast, but this is a video, so you can slow it down. We can talk about it some more. That's really something that is going to be a big part of the first couple chapters. Most of that stuff's in chapter 3. Just basic crosses: monohybrid cross, test cross, monohybrid cross, dihybrid cross, and then test cross. Okay, so we'll leave it at that. The main point of this was just to kind of show you how to go through the virtual fly lab. I got a little carried away, but hopefully you know how to do it now. And we will stop it there.